Hello students, myself Deepika Rana from DR PHYSIKA channel. Welcome to all in this channel. In today's video session, I am going to discuss about some basic information of acids, base, salt and indicator. So watch this video till the end. Let's start. So let us take the look at the properties of acids, bases and salt and how they differ from each other. So let us understand the properties of acid. So acids are sour in taste. Acid turns blue litmus paper to red and it releases H plus ion in an aqueous solution. Acid has a pH value lower than 7. H plus ions in the solution are responsible for a acidic character. Now let us look at the properties of a bases. Bases are bitter in a taste. Base turns red litmus paper to blue. It releases OH minus ion in an aqueous solution. Base has a pH value higher than 7 and OH minus ion is a solution are responsible for a basic character. Now let us understand about salt. Salt as expected have a salty taste. For example, NaCl that we eat taste salty. But there are some salts which have a sweet, sour or even a bitter taste. We know that the famous neutralization reaction in chemistry, acid plus base give us the salt plus water. Let's take one example of the neutralization reaction. HCl, that is hydrochloric acid, when react with sodium hydroxide, that is NaOH, gives us sodium chloride, that is NaCl, plus water, H2O. That means salts are formed by neutralization of acid and base. So a simple rule of the identify salts is that it should contain one positive ion other than H plus and at least one negative ion other than oxide or hydroxide ion. That means acid can neutralize a base and base can neutralize an acid. In terms of the taste, it means the acid can neutralize bitter taste of base and a base can neutralize a sour taste of an acid. Now let us understand about indicator. Substances which show the acidic or basic behavior of other substances by change in color and odor are known as an indicator. There are many types of indicators. Some common types of indicators are natural, olfactory indicators, synthetic indicator and universal indicator. Let us learn about indicators one by one in detail. Indicators obtained from natural sources are called natural indicators. Litmus, turmeric, red cabbage, China rose, etc. are some common natural indicators used widely show the acidic or basic character of substances. Litmus. Litmus is obtained from lichens. The solution of litmus is purple in color. Litmus paper comes into two colors, blue litmus and red litmus. Turmeric is another natural indicator. Turmeric is yellow in color. Turmeric solution or paper turns reddish brown with base. Turmeric does not change color with acid. Red cabbage. The juice of red cabbage is originally purple in color. Juice of red cabbage turns reddish with acid and turns greenish with base. Now let us learn about olfactory indicators. Substances which change their smell when mixed with acid or base are known as olfactory indicators. For example, onion, vanilla, clove, etc. are included in olfactory indicators. Paste or juice of onion loses its smell when added with base, and it does not change its smell with acid. Vanilla. The smell of vanilla vanishes with base, but its smell does not vanish with an acid. Now let us learn about synthetic indicator. Indicators that are synthesized in laboratory are known as synthetic indicators. For example, phenolphthalein, methyl orange, etc. Phenolphthalein is a colorless liquid. It remains colorless with acid but turns into pink with a base. Methyl orange is originally orange in color. It turns into red with acid and turns into yellow with base. Using a litmus paper, phenolphthalein, methyl orange, etc., only the acidic or basic character of a solution can be determined. But use of these indicators does not give the idea about the strength of acid or base. So, to get the strength as well as the acidic and basic nature of given solution, universal indicator is used. So let us learn about the universal indicator in detail. Universal indicator shows the different color over the range of pH value from 1 to 14 for a given solution. Universal indicator is available both in the form of strips and solution. And yes, universal indicator is a combination of many indicators such as water, propanol, phenolphthalein, sodium salt, sodium hydroxide, methyl red, etc. Now look at this table. This table shows the color change with red litmus paper, blue litmus paper, phenolphthalein and methyl orange solution for each of the sample solution. This color matching chart is supplied with the universal indicator which shows the different colors for different values of pH. Thank you for watching this video. See you in the next video lecture.